The single strand RNA genomes of viruses in class 4 and 5 are replicated by synthesis of their corresponding complementary strand. So the current complementary strand, they replicate with their corresponding strands, and they are then used as template for the new strand. Like if that is plus strand RNA viruses, so it will produce negative strand, and the negative strand will be then work as intermediate for the positive sense RNA. And similarly, if that is a negative RNA virus, that will produce positive sense RNA, and that will then produce, they will use as template for a negative strand, and the negative strand will be then encapsulated in the virion. So the synthesis of each RNA molecule requires recruitment of RNA-dependent RNA polymerase to the 3' end of the template. So the 3' end of the template will be used by the RNA-dependent RNA polymerase to produce the other the complementary RNA. And therefore, both plus and minus strand RNA must have a binding site for the enzyme at the 3' end. So at the 3' end, so there should be so on this strand there should be binding site for the RNA dependent RNA polymerase, and on this strand there should be the site for the enzyme. So that will mean when the 3' end is the RNA dependent RNA polymerase binds at the 3' end, it will make the other strand, and that will be from 5 to 3. So that will be from 5 to 3. So this will be 5. And the complementary of this will be this one. So that this site, even this site will be 3' end. It will be like this. It will be, if I draw it, if I will try to draw it like, if this is 5 to 3, if this is 5 to 3' end, sorry, and the other strand is and so that will be again, the other strand will be, this will be 3 and this will be 5. So that means if we have a sequence, if this is positive sense RNA, this is negative sense RNA, and what will happen? The polymerase will, like, that will bind, the polymerase will bind at the 3' end. The polymerase will bind here. So the RNA dependent RNA polymerase needs to, so that needs to, this RNA dependent RNA polymerase needs to have site on the RNA. So this site has this RNA has the site for the polymerase, for the RNA dependent RNA polymerase to bind here, and once it bound there, it will move in the direction, and it will move in this direction, and it will produce what? It will produce the negative sense RNA. Now the negative sense RNA has 5' end here. So that will mean when it is produced, it will have a binding site here. Sorry. It will have a binding site on the other side, on at the 3' end. So the 3' end here, there will be a binding site. So, and it will make transcription. It will replicate into this direction. So that means if we have an RNA, positive sense RNA, that means if we have a positive sense RNA, it will have the binding site, the complementary strand will have the binding site. That this will be the 3' end. So this binding site will be complementary to this 5 prime end. This will be 3 prime end of the other strand actually, and this will have so this will be complementary. This will be complementary to this, and this is actually the binding site. This is not the binding site, but this is actually the complementary sequence of that binding site. So the 5 prime end of the one sequence will be complementary to the 3 prime end of the other actually. You got my point or not? So simply, the synthesis of each RNA molecule requires the recruitment of RNA-dependent RNA polymerase to the 3' end of the template. So the 3' end of the template, the RNA-dependent RNA polymerase will be recruited that will bind there. And for binding, recognition is important. So the polymerase will recognize the 3' end. So that will means there must be some specific sequences at the 3' end of an RNA so that RNA-dependent RNA polymerase could recognize that sequence and could bind there for replication. And therefore, both plus and minus strand RNA must have a binding site for the enzyme at the 3' end. So 3' end of both plus and negative sense strand will have binding site for the RNA-dependent RNA polymerase. And next is class 4 viruses. These are the class 4 viruses. They of eukaryotes replicate their RNA in association with the cytoplasmic membranes. So these class 4 viruses, they do not replicate like we say they replicate in the cytoplasm, in the cytosol. They do not replicate in the cytosol, but they need specifically the cytoplasmic membranes. They replicate in association with the cytoplasmic membrane. They need membranes to multiply themselves. So for many groups of viruses, including Picornaviruses, these membranes are derived mainly from endoplasmic reticulum. So they are derived from endoplasmic reticulum, but other membranous structures are used as well, including endosomes by toga viruses, chloroplasts by tubemoviruses. So that will mean that this class of viruses what they will need the membranes, so the membrane structures, so they will need the membranes. So and in with these membranes they will bind, they will bind within these membranes, and they will multiply there. ठीक है, ये मेम्ब्रेन के साथ बाइंड करेंगे और इन मेम्ब्रेन स्ट्रक्चर्स में ये अपने डीएनए को, ये सॉरी अपने आरएनए को मल्टीप्लाई करेंगे. इज इट क्लियर अप टिल नाउ?
यस
तो
सर.
जी जी, कौन एक्सप्लेन करेगा इसको? इजी है. सो अगर हम इसको समझ जाए तो फिर हम आगे जा सकते हैं. जी, हु विल एक्सप्लेन? निम्रा शबीर इज देयर. निम्रा?
यस सर.
जी, इस स्लाइड को बता दें.
सर एक दफा पढ़ लो.
जी जी, प्लीज टाइम ले लें.
ओके सर. सर इसमें ये बताया है कि जो हमारे पास क्लास 4 और क्लास 5 वायरसेस हैं, इनके जो सिंगल स्टैंडर्ड जीनोम्स हैं, इनमें जो रेप्लिकेशन होती है, सबसे पहले इनके जो कॉम्प्लीमेंट्री स्ट्रैंड्स हैं इनकी सिंथेसिस होती है। फिर इनकी सिंथेसिस के बाद जो हमारे पास स्ट्रैंड्स आते हैं उनको हम लोग एज अ टेम्पलेट यूज कर सकते हैं, ताकि आगे जो न्यू जीनोम की कॉपीज हैं उनकी हम लोग सिंथेसिस करवा सकें। उसके बाद जो हमारे पास टेम्पलेट, जिसको हम लोग टेम्पलेट स्टैंड जिसको एज अ यूज कर रहे हैं, उसके 3' एंड पे हमारे पास आरएनए डिपेंडेंट आरएनए पॉलीमरेज जो है वो हमें चाहिए, ताकि एक आरएनए मॉलिक्यूल की जो है वो सिंथेसिस हो सके। तो इसी वजह से जो हमारे पास दोनों ही माइनस और जो प्लस आरएनए स्ट्रैंड है, इनके पास जो 3' एंड है वहाँ पे बाइंडिंग साइट होती है, और जो हमारे पास यूक्रियोट्स के क्लास 4 वायरसेस हैं, वो साइटोप्लाज्मिक मेम्ब्रेन के साथ एसोसिएट होके जो है वो रेप्लिकेशन शो करते हैं। तो हमारे पास बहुत से ग्रुप्स ऑफ वायरसेस हैं, जिनमें ये आगे एग्जांपल दिए हैं, पाइकोरोना वायरसेस। ये सब क्या है कि जो मेम्ब्रेन्स हैं ये बेसिकली ड्राइव होती हैं एंडोप्लाज्मिक रेटिकुलम से, और जो अदर मेम्ब्रेनस स्ट्रक्चर्स हैं उनको हम लोग उनको भी हम लोग यूज कर सकते हैं, उनमें क्लोरोप्लास्ट और एंडोसोम्स आते हैं।
जी ठीक है. And okay, so proceeding further, and viral proteins including RNA polymerases are bound to these membranes. So these viral proteins will also associate themselves with these membrane, these cellular cell derived membranes. And if you can see, during the replication of single-stranded RNA, both plus and minus strands of RNA accumulate in the infected cells. So that means if that is the class 4, the both the plus and negative sense RNA will accumulate in the cell, even though the virion is well composed of plus strand. And in class 5, both positive and negative, they will accumulate, even though the virion is negative sense strand, they contain negative sense strand of RNA. But during replication, both of them will replicate, that they will accumulate in the infected cells, but not in equal amount. This is important. In plus stranded RNA viruses, plus RNA strands will be in excess. This will be in excess. And for minus strand RNA viruses, the reverse will be in the negative sense strand will be in excess. And large number of vesicles during the infection, if you can see
सर
the double vesicle membrane that
एक्सक्यूज मी
they are
सर
produced. Gigi, please.
सर ये जो प्लस ट्रेंड है वो एक्सेस में क्यों होगा और नेगेटिव आरएनए क्यों कम होगा?
ये असल में इसके रीजन इस तरह सच नोन नहीं है, लेकिन ये इन्फेक्शन में पाए गए हैं कि इन्फेक्शन जो प्लस स्ट्रैंड वाले इन्फेक्शन हैं, प्लस आरएनए वायरसेस वाले, उसमें द
जी सर.
पॉजिटिव सेंस विल बी मोर, एंड इन नेगेटिव सेंस आरएनए वायरसेस नेगेटिव सेंस आर मोर. They are found like this.
ओके सर.
The mechanism is not known why they prefer, the cell prefer positive sense, and what is actually how one strand is more and other is in comparison is there, in that is less. And large number of vesicle form are derived mainly from the endoplasmic reticulum, and these vesicles is each vesicle is 200 to 400 nanometer in diameter and is bound by two lipid bilayers. So two lipid bilayers, it will produce two. So the virus, they induce these sort of double layered vesicles. They induce them in order to have the sites to replicate themselves. So they need home for replication, and these double layered membrane structures, they are actually the homes and the sites for these replicosomes. Am I clear?
जी सर.
Okay, and next is
सर ये
the
जो हमारे पास आरएनए वायरस होता है वो जो है लार्ज नंबर ऑफ वेसिकल्स बनाता है.
जी जी, ये असल में वेसिकल बनाएंगे सेल के अंदर, सेल के मेम्ब्रेन से, जो है लाइक एंडोप्लाज्मिक
मतलब
रेटिकुलम, यहाँ पे हम
एंडोप्लाज्मिक रेटिकुलम के अंदर जाके ना.
जी जी, एंडोप्लाज्मिक रेटिकुलम से जो लाइक मेम्ब्रेन्स जो हैं वो डिराइव करेंगे, सेपरेट करेंगे, कुछ जो हैं
जी
वो
सर।
एंडोसोम्स से बनाएंगे, कुछ जो है क्लोरोप्लास्ट से मेम्ब्रेन डिराइव करेंगे। लाइक टोम्बस वायरसेस जो हैं they derive membranes from the chloroplast. Toga viruses, they derive membranes from the endosomes. And other like Picornavirus, they will derive membranes from the endoplasmic reticulum. So
ओके सर, समझ आ
ये वहां से डिराइव करके डबल स्ट्रैंड लेयर्स बनाएंगे और वहां पे खुद को मल्टीप्लाई करेंगे उन मेम्ब्रेन के साथ। अपने प्रोटीन्स भी एसोसिएट करवा के और होस्ट के कुछ प्रोटीन्स होंगे, सो उसके थ्रू ये खुद को मल्टीप्लाई करेंगे। And next is the primers. How the primers for both positive and negative sense strand synthesis is the small protein VPG, virus genome linked protein, which is uridylated at the hydroxyl group of the tyrosine residue. So is this VPG will have hydroxyl group tyrosine residue, and the hydroxyl group of that tyrosine residue is uridylated. So it will bind the VPG will bind at this site, and this sequence is known as CRE, and that is the CRE is cis acting replication element. So this is cis acting replication element, and this is the part of actually the virus. And at this site, it will provide template of two adenines. So the VPG will bind there, and this red color, this is RNA polymerase. The RNA polymerase will bind with the VPG and will start polymerization from there. In front of this A, it will bind U, and by binding U with the VPG, this is known as uridylation, and the second U will be binded with that then, and this is uridylated, and now the amplification will start from here. And this is actually the start of the primer as a primer. This will work as a primer. So uridylation, and this is common in poliovirus RNA polymerase. By poliovirus RNA polymerase, it will occur. It can occur in polioviruses. Uridylation of poliovirus viral genome linked protein, and the virus RNA polymerase adds UU to a VPG using AA in the CRE as the template. So this CRE is cis-acting replication element, this one. So cis-acting replication element, and using, okay. And next is these reactions takes place at a cis-acting replication element known as CRE, located in the stem loop in the virus genome, and AA sequence within the loop forms the template for the UU synthesis. And there is evidence that UUVPG acts as primer for negative sense RNA synthesis, but it is uncertain whether the primer of plus strand RNA synthesis in the VPG is the VPG or a uridylated derivative. So अब ये नहीं पता कि पॉजिटिव सेंस में नेगेटिव सेंस के सिंथेसिस के लिए मींस कि ये पॉजिटिव सेंस है, तो नेगेटिव सेंस के सिंथेसिस के लिए तो VPG यूरिडाइलेशन यूज होती है। लेकिन पॉजिटिव एक दफा नेगेटिव सेंस बन जाए, उसका प्राइमर क्या होगा, VPG होगा या यूरिडाइलेटेड कोई और डिराइवेटिव होगा, दैट्स इज नॉट नोन येट, दैट्स नॉट नोन। एंड next is much of the RNA in the replication complexes is present as replicative intermediates. So the replicative intermediates are present there in the RNA. So during the infection. So when a cell is infected with a virus, with an RNA virus, one can observe the replication complexes, and there will be there will be many replication intermediates, and these replication intermediates is an association of RNA molecule consisting of templates of RNA. These will be the templates of RNA and several growing RNAs. So an RNA with several growing RNA of varying lengths can be a replicating intermediate. Thus, some RNA intermediates consist of positive RNA associated with growing negative RNA, means the positive will be a template and the negative will be growing, and that status is known as replicating intermediate, while other replicating intermediate consists of negative RNA associated with growing positive sense RNAs. So both can happen and both will go side by side, while there will be positive RNA and the negative RNA will be grow on them, and there will be negative RNA and the positive RNA will be growing by using that negative as a template. And also associated with the RNA intermediate are copies of RNA polymerase. So RNA polymerase will also bind with these replicative intermediate to produce the complementary RNAs. इसकी समझ आ गई है?
Yes, sir.
जी सर.
ओके. इसका कोई बता सकता क्या था यहाँ तक? Please, come on. Rimsha. Rimsha is here. Zunaira?
Yes sir.
जी, ये अभी तक जो हुआ इसके बारे में बता दें.
सर इसके अंदर उन्होंने बताया है कि ईस्ट वाली साइड की बात करें.
जी जी.
तो इसके अंदर उन्होंने बताया कि जो आरएनए इंटरमीडिएट्स है ना, उसके अंदर दो तरह की चीजें होंगी. मतलब वो रेप्लिकेशन कॉम्प्लेक्सेस के अंदर ग्रोइंग आरएनए होता है और टेम्पलेट आरएनए होता है. टेम्पलेट आरएनए जो है वो एज अ टेम्पलेट यूज होगा और उसके ऊपर लाइक अगर वो पॉजिटिव सेंस है तो उसके ऊपर नेगेटिव सेंस में ग्रोइंग आरएनए होंगे. इसी तरह जो है अगर नेगेटिव सेंस है तो उसके ऊपर ग्रोइंग आरएनए जो है वो पॉजिटिव में, लाइक जिस तरह वो पूरा बनता है स्ट्रैंड. और इसके साथ आरएनए पॉलीमरेजेस भी होते हैं जो कि दूसरे स्ट्रैंड्स बनाएंगे.
ठीक हो गया. ठीक है. And Noman is here.
Yes, sir.
जी, अभी तक जो पढ़ा है उसमें बता दें क्या आपको अच्छी समझ आई है. जिस चीज की समझ
सर
आई
मैं
है.
सर नेक्स्ट लेक्चर में सुना दूं.
क्यों अभी क्यों आपकी कोई काम है कोई?
नहीं
ये बिजी हैं?
नहीं सर बिजी.
अच्छा, अनीका आप बता दें. अनीका.
सर उनका बताया हुआ ना कि कैसे वो रेप्लिकेट करती है और आरएनए इंटरमीडिएट एज अ वो आरएनए इंटरमीडिएट जो होता है ना वो एज अ टेम्पलेट होता है इसमें. सर वो मुझे इसकी समझ नहीं आ रही. तो आरएनए इंटरमीडिएट है ये एज अ प्राइमर यूज होता है यहाँ.
अच्छा, तो और किस चीज की समझ नहीं आई? ठीक है, ये
बस
अच्छी बात है, ये डिस्कस करने होना चाहिए कि जो चीज अगर
बस
समझ नहीं आए तो. जी,
ये इसी की नहीं आई.
ठीक हो गया.
ये जो आरएनए इंटरमीडिएट है ना, मैं ये पूछ
जी
रही
जी.
हूँ ये प्राइमर के तौर पे यूज होता है
ये
यहाँ.
है होगा क्या? यू वांट टू आस्क दैट ना?
यस सर.
ओके, चलें इसको कमिंग स्लाइड में ये डायग्रामेटिकली एक्सप्लेन हो रहा है. अफान. अफान तारिक. अफान.
यस
ओके.
सर.
जी अफान, क्या कितना समझ आई आपको? क्या समझ आई आपको? अपने वर्ड्स में एक्सप्लेन कर दें. या आप भी नेक्स्ट क्लास में एक्सप्लेन करेंगे?
यस
या
सर.
जिस चीज की समझ आई है वो बता दें, जितनी समझ आई है वो बता दें. जी. चलें, रिदा है. रिदा. रिदा अथर. Rida is there? Maida, Maida? Maida is there or not? Okay, you raise your hand, you have problem with mic. Okay, chale, no problem. Fatma Bibi,
यस सर
जी क्या समझ आई है क्या नहीं आई? Let me know.
मैंने अभी ज्वाइन किया क्योंकि मेरा आ रहा था अभी मेरा ज्वाइन हुआ है तो मुझे नहीं पता कि आपने क्या पढ़ाया अभी तक फिलहाल
Okay, Rimsha Asim,
यस सर
जी आपको क्या समझ आई है?
सर इसमें जो है ना हम सिंगल स्ट्रैंड आरएनए रेप्लिकेशन के बारे में पढ़ रहे हैं कि रेप्लिकेशन में क्या है कि इनके एक स्ट्रैंड से उसी की कॉपी बन रही है हमारे पास। तो उसमें बताया हुआ है कि जो रेप्लिकेशन का जो कॉम्प्लेक्स होता है जिसमें आरएनए की कॉपी बन रही होती है उसमें जो हमारे पास समटाइम्स क्या होता है कि आरएनए जो है वो एज अ रेप्लिकेटिव इंटरमीडिएट जिसको आरआई से डिनोट किया गया है प्रेजेंट होता है। और ये जो आरआई है ये क्या है हमारे पास एसोसिएशन होती है आरआई की आरएनए के साथ और ये जो है एज अ टेम्पलेट के तौर पे एक्ट करता है। और इसकी फिर हमने बताया हुआ है कि यानी कि ये कॉन्सिस्ट होता है आरएनए पॉजिटिव विद ग्रोइंग नेगेटिव आरएनए यानी कि दोनों तरह से हो सकता है पॉजिटिव आरएनए की एसोसिएशन विद नेगेटिव आरएनए के साथ और जो है नेगेटिव आरएनए की एसोसिएशन हो सकती है पॉजिटिव आरएनए के साथ। और उसके बाद बताया हुआ है कि जो आरएनए है ये कॉपी होता है यानी कि हमारे पास आरएनए पॉलीमरेज के साथ भी उसकी एसोसिएशन होती है तो ये रेप्लिकेशन में हेल्प करता है।
ठीक हो गया रिमशा, ठीक है। And coming next is the diagram, and that is like in the blue color you can see there is this is an representation of a positive RNA, and this is a positive RNA virus actually. On the 5' end you can see two uracils, and then there is at the 3' end you can see multiple of like A, and the VPG is bound at the 3' end, and the RNA dependent RNA polymerase is also bound there. What it will do, it will make one of the uracil, then another uracil, and it will scan the whole RNA then, and going from 5' to 3' direct in the 5' to 3' direction using the template from the 3' to 5'. And once the other strand is produced, so what will happen, this will like scan and it will go on to the RNA using that template, and that was actually positive sense, and now it is producing the negative sense strand. When it reaches here, so now here at this stage you can see like one polymerase will move on scanning, the other polymerase will start amplification, and they will move, that will move in that direction. So this is replicative form. This is a replicative form. So in the replicative form, the plus strand in association with negative sense strand, you can see that in association with RNA dependent RNA polymerase. So all of these will be present there in the intermediates. Similarly, when these negative strands sense strands are produced, it will be used as a template for the positive sense strand. Now the time for production of the blue color positive sense RNA production is required. Now you can see now it will produce and the VPG UU will bind there at the 3' end. Now this is the 3', this is the 3' end. It will bind at the 3' end, and it will scan in the 5' direction of the template, and now it is producing the positive sense, and also it is producing another sense positive sense strand. Now one of the strand we will call that as will be used as messenger RNA, the other RNA will be used as progeny RNA, and this progeny RNA and the third type of the RNA will be used to produce the negative sense RNA again. Now, check this one, the replicative intermediates. It contains the positive RNA for the genome to assemble, for messenger RNA to produce proteins, for the template as a template of positive sense to produce the negative sense RNA. So this is how when a replication starts, it employs different sort of replication mechanism and functions actually. इसकी समझ आ गई है?
सर लास्ट वाला आप दोबारा एक्सप्लेन कर दें, लास्ट वाला पॉइंट.
Okay. So here, if you can see, this is now here the negative sense strands are produced. Once the negative sense strand is produced, it will be used as a template for the positive sense strand. So the positive sense strand, so now there are 2 types, even 3 types of positive sense strands that are producing. The first one is will be used as progeny genome. It will encapsulate in the virus. The other positive sense strand will be used to used as a messenger RNA. Messenger RNA means the polymerase will work on it and will produce proteins in the cell. And the third one is used as as a template for producing negative sense RNA viruses. Am I clear?
Yes sir, thank you.
Who will explain this? Gigi, who will explain this? Rida, जी.
सर ये जो पहला स्टेप हुआ उसके अंदर आरएनए के पॉजिटिव जो आरएनए है उसके ऊपर आके अटैच हुआ है सर, ये जो आरएनए पॉलीमरेज अटैच हो रहा है ना फर्स्ट स्टेप के अंदर जो 3 प्राइम पे अटैच होगा एंड देन आगे से नेगेटिव आरएनए बनेगा. सर जब रेप्लिकेटिव इंटरमीडिएट बन रहा है तो वहां पे 2 क्यों अटैच हो रहे हैं?
Okay. So actually it is like when when there is an RNA, one RNA, the polymerase will bind, but that will require VPG as a primer. This will require VPG, virus genome linked protein, as a primer. Wait for a while. Sorry. So this is a positive sense RNA. Now the polymerase will bind at the 3' end, but it will require primer. In this very case, the primer is not an RNA. It will be viral genome linked protein. It will be a protein. So we described earlier from this slide, we described earlier like how the VPG will be used as a primer. The polymerase will add first uracil and then the second uracil, and that will mean the uridylation of the VPG. So and then it will start the polymerization then. So in that case, it will start polymerization and will produce the negative sense strand. So now while producing this strand, this area is now open, and this will be open, and the next VPG will bind there, and the next polymerase will bind there again, and will start replication of the RNA, of the same RNA. And this is common for eukaryotes as well. While we have RNA in the cell, we know that while we have messenger RNA out of the context of viruses, a messenger RNA, it doesn't like only one polymerase will scan this. It's not like that. Not only one polymerase will scan, this polymerase will scan, it will scan the RNA, And meanwhile, another polymerase will be bound there. Sorry, will already be bounded there, and it will have polymerized. It will have polymerized the sequence. It will have polymerized the sequence, this one, and there will be one another ribosome, one another polymerase, like here, and like here. There will be a third polymerase, and the polymerization from here will also. So all these polymerases will go in this direction. So this direction, and this will go in this direction. So, and same is the case with the messenger RNA. Messenger RNA, when there is messenger RNA, multiple type or multiple ribosomes, they bind there, the ribosome bind there, and they scan the RNA. They scan the RNA in the direction, like here in this direction, I suppose in this direction, and they will produce proteins. Each of the ribosome will produce protein, and they will scan on the same RNA. So this is an efficient way of producing, of giving the yield actually. So like our concept before were like we were thinking that maybe only one ribosome per RNA or one polymerase per RNA is there, but that is not the case. And this status is known as polysome. Multiple of ribosome, they bind on the single same RNA. And similarly, in the case of viruses, multiple polymerases, they will bind there in order to efficiently producing many RNAs on the same time and space. So it will produce many RNAs. So the single RNA will produce multiple negative sense strand RNAs, and these multiple single stranded RNA will be producing multiple positive sense RNAs, and these multiple positive sense RNA, some of them will be produced will be used as progeny for encapsulation, some will be used for messenger RNA to produce proteins, and some will then again used as template for producing negative sense RNA to like recycle and to again start the cycle producing the positive sense RNA. And iski samajh aa gayi hai? Every person
Yes,
is
sir.
clear or not?
Yes,
Who
sir.
will explain? Who will explain this diagram?
So मैं कर
Come
देती हूँ।
on, who will explain? जी बताएं।
सबसे पहले हमारे पास फर्स्ट डायग्राम के अंदर बताया उन्होंने कि पॉजिटिव सेंस आरएनए स्ट्रैंड है, और हमने पीछे बात की है कि पॉलीमरेज जो है वो 3 प्राइम एंड पे हमारे पास आके अटैच होता है। इसके साथ वायरल प्रोटीन लिंक जीनोम है, वो एज अ प्राइमर यूज हो रहा है, वो 3 प्राइम एंड पे आके अटैच हो रहा है। उसके बाद 2 यूरिसिल कोड की है जिसको हम यूरिडाइलेशन कह रहे हैं। उसके बाद इस पॉलीमरेज ने आगे पूरे स्ट्रैंड की पॉलीमराइजेशन करवानी है और एक नेगेटिव सेंस स्ट्रैंड बना देना। उसके बाद सेकंड में जिधर रेप्लिकेटिव इंटरमीडिएट दिखाया है, वहां पे उन्होंने दिखाया कि एट अ टाइम जो 2 हमारे पास पॉलीमरेज वर्क कर रहे हैं, जिसे हमने पोलिसोम का भी नाम दिया है कि एट अ टाइम डिफरेंट मतलब मैसेंजर आरएनएस बन रहे हैं। उसके बाद थर्ड में हमारे पास उन्होंने वही जो नेगेटिव स्ट्रैंड बना था उसको दिखाया हुआ है कि 3 प्राइम एंड पे आके दोबारा हमारे पास पॉलीमरेज की अटैचमेंट हो रही है और यूरेसिल कोड हुए 2 मतलब वीपीजी प्राइमर लगा है और आगे पॉलीमराइजेशन करके एक पॉजिटिव सेंस आरएनए स्ट्रैंड बनेगा, जो अब फिर दोबारा पोलिसोम वाली बात आ रही है यहाँ पे कि डिफरेंट स्ट्रैंड्स बन रहे हैं एट अ टाइम। और एक पहले जो बन रहा है एज अ प्रोजेनी जीनोम बनेगा जो कि असेंबल होगा, आगे इनकैप्सुलेट होगा वायरस के अंदर। एक मैसेंजर आरएनए बन रहा है जो आगे प्रोटीन बनाएगा, और एक दोबारा ऊपर उन्होंने दिखाया हुआ है कि उसी से पॉजिटिव जो नीचे बना है आरएनए स्ट्रैंड, उसी से दोबारा नेगेटिव सेंस आरएनए स्ट्रैंड प्रोड्यूस होगा।
ठीक हो गया। मोइज?
Yes, sir.
Okay, do you want to say something?
No, sir. Sir, मेरा इंटरनेट नहीं है स्टेबल, तो इस वजह से बार-बार रिकनेक्ट
Okay. Okay. So for today, I think it is enough, and in the next class we will start actually. okay, like if we conclude this, the poly A sequence at the 3' end of the plus strand is not synthesized in the usual way. Like if you can see the 3' end of the plus strand, if this is plus strand, this will be plus strand. The 3' end of the plus strand is not synthesized in the usual way. It is transcribed from a poly U sequence. It is transcribed from a poly U sequence at the 5' end of the minus strand. If this is the minus strand, so the 3' end, so this is the 5' end, sorry, this is the 5' end, and the 3' end will be used from this here, from the 3' end will be from here. So, and 5' end of the minus strand. So this will be the 5' end, and this will be the 5' end of the minus strand, and this will be the 5' end of the plus strand. So this will be from the poly U sequence of the negative sense, and it has been estimated that in a poliovirus infected cell, 2,500 RNA molecules are synthesized per minute. So you can imagine how fast is the RNA multiplication. 2,500 RNA molecules are synthesized per minute, and the virus controls RNA synthesis in such a way that about 50 times more plus RNA than minus RNA is made. So previously we were of the opinion that bacteria, they multiply, and they multiply at in every 30 in every minute they multiply. So here you can see 2,500 RNA molecules are produced per minute, and the virus controls RNA synthesis. In plus stranded RNA, so the positive RNA will be 50 times more than the negative sense RNA. And some of the positive RNA molecules are used as template for further negative RNA synthesis. So some of them will be used for synthesis of the positives, for synthesis of negative sense RNA. Some of the positive will be used for the synthesis of negative sense RNA. Some will function as messenger RNA, and some are distinct to be the genomes of progeny virions. So here is the case. So some will be used as progeny genomes. Some will be used as a messenger RNA, and some will be used to produce the negative sense RNA. And here the 3' end of the genome of the plus strand is synthesized from the poly U sequence of the 5' end of the minus strand, and that's it.
तो यानी सर ये बैक्टीरिया से ज्यादा जल्दी रेप्लिकेट कर रहे होते हैं वायरसेस?
Exactly.